ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. Very good morning to all of you. It's a pleasure to introduce our guest speaker today, Dr. Ravichandra Venkateshappa. Dr. Ravichandra is a postdoctoral at Cardiovascular Institute at Stanford University. His ground breaking work focuses on understanding electrical activity of the heart with a specific interest in how ion channels function and how they can be targeted to treat heart disease. His research has been published in numerous prestigious journals, including the Journal of the Royal Society Interface and Stem Cell Research. Today, he will be sharing his thoughts on from skin cell to solution, how iPSCs accelerate drug discovery. Before starting his talk, I want to tell you that he is a alumni of Vijaya College and student of Mahesh sir. I forgot to introduce myself. I am Suresh, uh, Joint Secretary of Prajna Foundation and also biology NEET educator. So over to you, sir. Please take over the session.
Krish, it's Mahesh, right?
You can start.
Me? Okay, yeah. Thanks for your kind words, Suresh, and also thanks for inviting me to give a talk for this foundation. So I'm not gonna be giving a talk. I'll just give a brief overview of how I made it to research that I'm currently doing. Just I'll share my journey and some of the work that I'm currently doing at Stanford. So I'll start, with I have made some points that I want to discuss. I'll just start with my research journey. So as Suresh mentioned, I was an alumni of Vijaya College, where, you know, I think that's where I learned the being innovative or being inquisitive about things. I had teachers like Shafi sir and the late GMN sir and also Mahesh sir who taught me biochemistry. So this is where my interest in science grew, and then it became, then I decided after moving up, graduating from Vijaya College, to do masters and then do PhD, and then currently I'm doing postdoc. So just to give a brief background of where I come from. So I come from a very small village in Kolar district. It's called Rajakrishnapura. It's close to Alambadi, Chikkatirupati. And then I started my schooling there. And from there, I went to Bangalore, and where I studied again in a government school, Puttainhalli Government High School, primary and high school. So where I did up my seventh grade, I finished, and then I went to Model Education Society in Jayanagar, where I did my 10th class, and then I went to Vijaya College. I've been associated with the Vijaya College for a very, very long time. So I did my first plus 11th grade, 12th grade, and BSc all in Vijaya College. So I have a lot of gratitude and also love for Vijaya College, and also all the people who are associated with the Vijaya College. Maybe we can have it more interactive. Anyone has any questions, you can just stop me and ask me questions. I'd be happy to answer and also discuss the things. And then I will, so now I would try, and maybe I can describe how I got interested in research. So it's not that even when I was a child, like fifth grade, sixth grade, I was very curious about things. So I still remember one of the teachers from my school, me touching the microscope for the first time, and then I remember the memories of my teacher who showed me the cells for the first time. I observed the plant cells, and those are the things that really seeded the interest in research. So that's how I decided that I want to become a scientist. and then I really, and also I would like to mention that, I studied in a Kannada medium school. So for me, it was really, really uh, very, for me, it was an unreachable goal, because being a government school student and also like studying in Kannada medium, and then up to 10th grade, I studied in Kannada medium, and then I had to transition to an English medium if I had to take science. And that's where I joined Vijaya College, um, where it was the first initial days were really, really tough. I even like failed in like some of the initial test. But later on, I really worked hard because I had like a passion for science. My only goal was to become a scientist, so I never thought of becoming anything else. So I wanted to pursue that career in science. So I knew that if I had to pursue my career in science, I had to really work hard and get good grades and then go to good colleges. So with that, motivation or goal in mind, I really, really worked hard during my 11th grade, and I got like reasonable good grades during my 12th grade. And also like, I cleared my all my CET exam. Then it was called, now it's called NEET exam. Then it was called CET exam, Common Entrance Test. I did clear like engineering and medical college entrance exams, but still I chose to go for BSc or Bachelor of Basic Sciences because of my interest in basic science. So during Vijaya College days, I was very active in the lab. That's why many of, including my sir, and many of the teachers or the lecturers still remember me because I was so proactive, especially in the lab. And so the good thing about being a Vijaya College alumni or studying at Vijaya College was, the teachers. I had like this very, very good, motivating teachers. So whenever I went to teachers, I know our Indian system. It's not like a research oriented, academic pathway. Sorry, academias. But still, my teachers there in Vijaya College, whenever I went and I asked them, hey, I want to do this research, I want to do this experiment, they never said no to me. So I used to be, very active in chemistry lab. I used to like so many different things, and they were like, including the lab attenders and the professors were really, really encouraging. And then had great teachers like GMN sir, who really, really changed my life. I mean, I can say my life because he got me to in contact with one more person who was a Harvard professor. He went to Harvard, and then he was leading a company there. And through him, I learned the way of doing good science. So even during my undergraduate studies, I did do some experiments. I don't know if Maya sir remembers. I used to work on this enzyme kinetics, by designing new apparatus and all those stuff. So, the reason that I'm telling you this, the people who are listening to me, is if you have the goal that you want to reach, the and if you surround yourself with the good people, they will, if they see you that you really, really want to reach that goal, then everyone would be like with you. They will help you to reach that goal. And that's what happened with me. So I finished my bachelor's, and then I wanted to study biochemistry. And then I went to Central College, where I got like, again, I was not the topper, but I still like got I was able to clear masters. And then after masters, I went and I joined Biocom, Syngene International, where I got a chance to work on like the actual application oriented research. So there I spent 4.5 years, and then I decided that, as I said, my goal was to become a scientist. If I were to work in an industry, I would have stuck myself, to industrial research, which was not bad. But again, I wanted to do like something that's like out of the box. So if you want to do like something that's out of the box with your own ideas, industrial setup doesn't allow won't allow you that. So you have to have academic research labs to do those out of the box thinkings and testing new ideas. So that's what like motivated me, and then moved for my PhD. I moved to Canada, and then so during my PhD, what I did is, so when I was in the company, I learned about electrophysiology. So electrophysiology is basically where we use, some of the combination of physics, and to understand the electrical behavior of living cells, which are, sorry, got a message here. So electrophysiology, so I started learning electrophysiology when I was in the company, Biocon. And then when I wanted to do PhD, I decided that this is the field that I wanted to, pursue because it was so interesting. So my first instance of, learning about electrophysiology was when I was a master student or final year of BSc. That's when I learned the neuron physiology and those action potential recording and using that, tiny pipette, and you just touch the cell, and then you could see these changes in the membrane potential. That really triggered me, and me and my friend, one of my good friend Wachan, we even thought of building electrophysiology rig. Then we didn't know what it was, but still we wanted to build that rig. So that's how I got really interested in electrophysiology, which was so cool. You can put a very tiny electrode onto the cell, and you can record the changes in the tiny membrane potentials. And also, like, you can record how these membrane proteins that are sitting on the cell membranes, they allow ions to move in and out of the cells. So that's why I decided, okay, I'm gonna go to this PhD, and I'm gonna do PhD in ion channel electrophysiology. In addition to that, so the most hot topic right now, and even like 5 years ago or 10 years ago, in especially in the medical biomedical sciences, is stem cell technology. So stem cell technology is, like, I was really fascinated. So again, I learned about this when I was in the company. So what you can do with the stem cell technology is, so we know, like, there are two kinds of stem cells. Like, you can get the stem cells from like human embryos, and or like, ethically, you will have ethical problems and other stuff. But there was a technology that was, a professor by Yamanaka, who got a Nobel Prize in 2016. So he invented a technology where you can add like 4 transcription factors, and you can convert any adult cell back into like a pluripotent stem cell. So that stem cell again can be driven to any kind of cell that you want. Like, say you want to make heart cell, you want to make a neuron cell, you want to make a muscle cell, like any kind of cell that you can virtually think of. You can drive these stem cells into that kind of specific type of cell. So when I was in the company, I saw, okay, people were making these iPSCs to neurons, iPSCs to cardiomyocytes. That really fascinated me. Then I thought, okay, I have this electrophysiology that I want to do, and then I'm gonna combine this stem cell technology, which was very hot then. And then I moved to a lab where this professor was working on iPSCs in and using ion channel electrophysiology using iPSC technology. So, and so the next point I want to talk is, like, so far, like, does anyone have any questions or anything?
We
Okay,
can take up at the end.
okay, yeah, okay, Suresh. So my PhD, as I said, now I was in a lab where we had these iPSCs and ion channel electrophysiology. So then I thought, okay, so my PhD was on one of the cardiac diseases. So if you know the stats, after one of the major leading cause of death is cardiac, diseases, cardiovascular diseases. They are the leading cause of death in the world. So the specific disease that I worked on was called long QT syndrome. So this long QT syndromes is basically, what happens is, because of the mutations in some of the ion channel genes. So the cardiac cells, so I'll give a brief idea of, like, people who don't know what are ion channels and other stuff. Ion channels. So these ion channels are basically membrane proteins. They form channels. They sit on the membrane. They form channels, like basically the pores, and they allow potassium ions or charged ions to move in and out of the cell. And this movement of ions creates membrane current and leading to membrane potential changes. So in our body, like, majorly the electrically active cells, or like, the muscle cells, the movement of my muscle and my brain thinking, the neurons, and the cardiac cells. These are like the three major types of cells that are electrically active. So you might have seen, like, whenever a patient goes to a hospital, the first thing they do is they put on like the contact pads, and they look at the ECG of the patient. So ECG basically tells you the average electrical activity of the heart. So if a patient has any electrical abnormalities, heart problems, you can detect using ECGs. So this long QT syndrome patient, this ECG has a specific waveform, and we define those waveforms as P, QRS, T waveforms. I don't want to get into the details of those. It's basically, it's each cardiac cycle has this, typical waveform that remains same for all the humans. And these waves have a specific fixed time points. Say, as I said, there are specific waveforms, and this wave, the P wave, happens at one specific time, the duration of the P wave, and the T wave happens has a specific duration. So this PTV wave, PQRST waves, these waves basically nothing but, we have labeled the contraction and relaxation of the heart. So heart contracts and pumps the blood, right? So that those waves electrically we label them, and we see a specific waveform, and we label them PQRST waves. So patients with this kind of, with a long QT syndrome, what happens is these patients inherently they will have mutations in some of the ion channels that are that are responsible for forming this electrical activity, the PQRS waves that I said. So if there is a mutation that disturbs or makes the proteins, these ion channels, dysfunctional, that causes the prolongation of these waves, the Q wave or QRS wave or T wave. This will be like much more longer, or they can be shorter. So that causes disease. So I studied a specific disease where these patients had a specific mutation in one of the potassium channels. So that these patients channels, they were not opening properly. I can, in a layman terms, I can say that they were not opening properly. So there is a less potassium moving out of the cell. So because of that, what happens is, generally say, if the QT, the wave duration that I'm saying, the contractile cycle, generally takes like, in men it's around 450 milliseconds, in women it's 470 milliseconds. And because of this mutation, what happens is this 400 milliseconds can be extended to 500, 600 milliseconds, in some cases, like extreme prolongation. Because of this slower relaxation, QT prolongation time interval, these patients are at risk of developing sudden cardiac death. So, as I said, I have a normal heart. I can exercise, I can run. My heart adapts to the changes in my physical activity, and then it changes the rate. Whereas these patients with this kind of mutations, when they exercise or hear loud noise, when their heart rate goes high, they cannot cope up with the increased heart rate, and then suddenly collapse and they die. So how we can treat these patients? So one of the ways currently what we do is we just give them a specific type of drug that suppresses the sympathetic activation. You need to have a sympathetic activation to increase the heart rate, to depend to satisfy the energy demand when our physical activity increases. So they use beta blockers and they suppress this sympathetic activation of the heart. That's one way. But still patients experience sudden cardiac death. That's because these patients are not completely protected with the current medications. And also, how do we study the disease? It's very hard to get patient samples. If you have a cancer, you can take the tumor and you can understand the pathophysiology and see like what's happening, and you can add different drugs, and you can try to understand the disease mechanism and also see like which drugs work for these patients. But if they have a inherited heart problem, it's very hard to understand the pathophysiology. So that's where the stem cell technology comes in. So what we can do now with the stem cell technology is that, say if a patient has a problem, say I have a heart problem, inherited heart problem, say I have long QT syndrome. What we can do is I can go to the lab, give my blood. They will take my blood and they will isolate specific type of cells. And those cells, we can add like, some 4 transcription factors, we call them MNACA factors. And those transcription factors will reverse these cells back into stem cells, pluripotency state. So now I have my own stem cells in the lab growing. So what I can do is we can drive this. If I want to understand the cardiac disease, I can drive them through different chemical pathways turning on and off by using small molecules or through viral transductions. I can make those stem cells to beat like cardiomyocytes. So that's basically I have or the lab I have my own heart cells beating in the lab. So you can appreciate the technology. So if a patient comes, a patient's own heart cells you will have access to own heart cells where you can understand the disease, you can test the drugs, specifically for that patient. So in my PhD, what I did is, okay, we had this, a problem for a patient who had like this long QT syndrome, and we took the cells and we convert them into stem cells, and from the stem cells we made these beating cardiomyocytes. Then I used electrophysiology, we call like different techniques, and then what we identified that, so this mutation actually making these channels to close very slowly. So I'll just give a brief idea of these channels can be like closed or open or inactivated state. There can be in different states. So these channels, because of this specific mutation that I studied, these channels were like not closing properly. So they were opening for longer duration of, sorry, they were closing very faster because they're not allowing the potassium to move. So what I did is my PhD is like I took like several drugs, medicines, and you can imagine like I have like these patient cells and from the patient. So we use the cells from the patient, and then we understood like, okay, the potassium channels are dysfunctional. Next, like we wanted to like correct the dysfunction. So we tested like several drugs on these cells. And I would like to reiterate that, the cells are patient's own cells, and we tested the drugs on directly on the patient heart cells. And then we identified, okay, one of the drugs that seems to be like working. So it is like a preclinical study. So in the future, maybe if someone is interested, they can study the drug mechanism more, and they can take it to the clinical trial for patients. So now I'll move on to like my postdoc experience here. So currently my postdoc work, again, I'm working on stem cell technology. So the lab that I'm working on is like the world's leading stem cell technology lab, and my PI is like one of the world leader in iPSC technology and using iPSCs to understand the diseases and also drug discovery. Again, here what I'm doing is I have 4 patients who have this a type of inherited arrhythmic disorder. So these patients, they cannot exercise because if they exercise, they will die because there is a mutation in one of the calcium handling proteins. So these patients are at very high risk of sudden cardiac death. So if they exercise or even they by hearing loud noise, they collapse and they die. So you can see like how fatal the disease is. You can imagine how fatal the disease is. So these patients don't have any treatments. Again, they will be put on beta blockers just to suppress the sympathetic activation. Currently, what I am doing at in our lab is, so again we use take the blood from these patients, convert them into heart cells, and using heart cells, we understand the disease mechanism. And then I have added one more step here because I'm at Stanford and it's a big lab. We have like all the facilities. And now the biggest booming thing is AI, artificial intelligence. So what we are doing is we have a set of AI experts. They can use AI and come up with new drugs. So the things that used to take like years, 10 years, 20 years previously, because of the AI and stem cell technology, the drug discovery process has become like much more smoother and much more faster. So it generally takes now maybe around 5 years to get a drug to the market. So maybe in the future, maybe in 3 years, we'll have a drug in the market. And currently using AI, there are many drugs which are already in clinical trial in just 3 years. So my project is, okay, I understand the disease mechanism. Then I use this computational, AI modeling and other stuff to identify specific drugs that can work for these patients. So we call it like personalized medication, personalized or medicine or precision medicine. So we use this computational techniques and stem cells, combine them, and then we test the drugs, and we try to identify the drugs. And if you have any drugs that work, then we take them to the animal models, And in animal models, we test the drugs to understand that what they are doing, like at a whole, systemic level, to if they are causing toxicity, or if they have any other problems. So we'll study all this, or the what drug is doing to the body. We call it like our what drugs or what body is doing to the drug. So we try to understand all these things using mice models. And then once it works in mice models, then we'll go to higher animals, and then we go to the patients. So currently I'm at this stage where, at Stanford, working on, this 4 patients, and trying to understand the disease and trying to come up with a new medication for these patients. So Suresh, yeah, maybe I can stop here. I think it's, if you guys have any questions, I'm happy to take questions.
It's a request to participants to ask some questions related to that. If you have any doubts, so he's ready to ask. So he's ready to give answers.
So you were saying about some patients with inherent genetic related disease, right? So I think majority of the people won't get tested at all. So they might not even know that they have that risk. So what message do you have for such patients who don't even know that they have that disease?
I mean, that's the problem. That's why I think if you see in especially in countries like India, where still we are, developing country, so there it's a huge problem. But it's not a problem in these countries, like, Western countries, where say even like one incident of, a patient, a kid experience, syncope or sudden collapse, they immediately go to the doctor, and doctor diagnoses the patient, and they ask, do for DNA sequencing. When you sequence like a set of genes, or you can do like whole genome of sequencing. So those technologies are like very widely used here, as far as my understanding, I'm aware of. So using those DNA sequencing, we can identify and we can tell the patient to be, careful, and also we can then immediately put them on the drugs. But countries like developing countries like India, so the problem is, if you see, including Punit Rajkumar, it's my just I'm just, assuming that they may have like some kind of inherited mutations in them. It's like, oh, the person will be so fit, and it just suddenly collapses and, passes away. That's because they are not diagnosed. So diagnosis is a problem, because most of the cases, the first symptom is death. But you have to keep an eye with, these patients. What happens is the doctor would look at the family history. Say, if I have a kid and a kid experiences syncope, and suddenly if you go to the doctors looks at the kid, and then they go to for the sequencing, they would look at your family history, if there were any sudden cardiac deaths. That's how they can track the disease and be, make the patient, be aware of, these things. Probability, they have a higher chances of, cardiac death, or so. It's diagnosis as is the real problem. But
Yes.
it's getting there. People are like frequently visiting the doctors, understanding, their health is the key. They should not avoid, if they have like, history of their family, father dying of cardiac death, grandfather dying of this, or, young, especially young people, like from 30 to 50, if they have any sort of, death, sudden cardiac death, unknown, without any disease, that's where, people have to be cautious. The entire family has to be screened, go to the doctor, has to be screened, and see if they have any, this kind of inherited mutations.
Okay, thank you, and thanks for the session. Actually, it was very good. You made it understand simple. See, I'm not new to this term. I mean, I'm new to these terms, but you explained it in a layman language, and I really appreciate that.
Okay, thank you.
Thanks.
Yeah.
Ravichandran, I may know the reasons for the inherent the mutations, the factors influencing the mutations which leads to such.
I mean, we don't know the cause, like, because it's inherent. We don't know I don't know the cause, because it's through the family, it's passed on, it's hereditary, as I said. What caused those mutations? We don't know.
It cannot be known.
No, there is no way we can know like what causes those mutations. No.
So any change at the DNA level may lead to this?
Not all the changes, because some mutations can be just a benign. That's where the problem comes. Even if the doctor gets, if a patient gets diagnosed as, he has this mutation in this specific, channel, but knowing that whether that mutation causes disease or not is still a challenge. We don't know, because not all the mutations can lead to death. So it's a challenge, but still, irrespective of the mutation identified, doctor would put you on beta blockers, like on medications, because we don't know what mutation can cause death, what mutation cannot cause death. So one of the ways that we are trying to address that issue using stem cell technology, as I said, so we can take the blood samples and study the disease, see if it actually like causes the phenotype in vitro in the lab. Most of the cases, we can translate that to the patient.
So I have a doubt. Is that research is going on in India also? Have you ever been through it?
Yes,
Some-
yes, yes. NCBS is in STEM. I mean, Indian government has started Institute for Stem Cell and Regenerative Medicine. So they are the leaders right now. And there are other institutes, maybe Manipal. I don't know other institutions, but in STEM is like the leading, body, that leading institute in India, which are primarily focused on understanding using stem cell technology to understand cardiovascular diseases. And there is one of the big names is Dr. Dandapani. He's been doing like very good research on this, myopathies, especially as South Asians, we are more prone to a specific type of cardiac problem called myopathies.
Okay.
So yeah, I mean, government is investing on this, but still we are still like, very, very infancy stage compared to where we are at now at Stanford.
So any more questions that participants want to ask with Dr. Ravichandra?
Yeah, myself Nagaraj Patil. Thanks to Prajna Foundation and thanks to Ravi for sharing the screen and sharing all his insightful research studies. I have a question here. What is the ratio of appearance of this particular syndrome in regarding this population we have right now?
That's a great question, Nagaraj. So recently, I mean, I can say like for India, from the Indian context. So recent whole genome sequencing of 10,000 patients. Is it 10,000 or 1,000 patients they published? Sorry, I don't remember the exact number, but they showed that the frequency can be like 1 in 1,000. So that means if you take like all world average, so these mutations are like 1 in 10,000. That means 1 in 10,000 births may have this mutation, these kind of channelopathies. And there are certain channelopathies, we call them channelopathies or mutations in ion channels. There are certain diseases or mutations that can occur like 1 in 2,000. So even if it's 1 in 2,000, you can imagine from Indian population context, right? It's a huge population. Even 1 in 10,000 is like millions of people. So the frequency, depending on the type of the mutation, is like 1 in 10,000, 1 in 2,000. It can be 1 in 11,000, 1 in 20,000 as well. And some diseases are like very rare, 1 in like 100,000. So this entire group of diseases are called rare, diseases, because of, they're very rare. But in India, rare is not small, because even the rare, it becomes millions of people.
Yeah, thank you for the information. And how better, like as in Indian scenario, in what best way we can find out these particular syndromes? Is there any way for any
I think,
common man can?
yeah, I think many of the deaths, I mean, even I'm from India, right? Especially rural areas, many of the deaths are not even reported, Nagraj. So I think the first thing would be like reporting the deaths would be the first one, and autopsy, and keeping like the records of why the patients die. That's very important, because even after death, you can take the samples and do a sequencing and identify like why the cause of the death. And then we can there may be database, but I'm not aware of those database. But having these records would really help us to track back and tell the family, hey, you have this problem. Now tell the people that you have this problem. So the first thing would be getting access to, even from the rural parts of India, giving access to the basic medical services, and making people aware of the genetic diseases. Say, I know stories like, people just suddenly die, and then they say, oh, he was sleeping and died, passed away. And then I don't know, many of the cases, they don't even do like post-mortem of these patients or the deaths. So it's very important that, they go through the medical procedure, and then make people aware of these things. I think most of the people don't even know that these kind of diseases exist. So the first thing is making people aware, okay, these kind of diseases exist. If someone dies of like without, for no reason, suddenly, like especially young people, especially I see news, 23, 40, 30, 35, age people dying in India. So especially this kind of people has to be, thoroughly examined even after death, to identify the cause of the death. That would help us to understand, or give better health care for these patients. So as I said, I can summarize: making people aware and giving medical access to all these patients.
Just I want to add, Ravi here. Recently, just 2-3 days back or 1 week back, just the student who's studying in third standard,
Yeah.
boy, just collapsed and he died due to cardiac related problem.
Yes.
So might be this is the reason where we cannot predict, which particular age the heart attack or heart related problems will come.
Yeah, I mean, that's a good point. See, that's what I'm telling you, right? Many of these diseases, they start showing symptoms around 12 to 13 years of age. And sometimes, depending on the conditions, you may see like very young age people. So here we can just speculate that, it could be because of some mutations or some kind of inherited condition, but we don't know. That's why the kid has to be examined, and then they have to screen the family as well, so that, these things doesn't happen again. If they have like one more kid, if they have a father, mother, and their parents, everyone has to be, screened for these kind of mutations or causes of the death. So maybe if you know the family, I would suggest talk to the family and ask them to go to and see a doctor, and also ask them to identify the cause of the death for this kid. That's very important. So any other questions?
Am I audible?
Yep, yep.
Yeah, you have explained very neatly. Like, I have a question here. Like, how can a student choose this cardiovascular research from India? As you have said, like, okay, you have chosen Canada as your doctorate study, and after that, now you're in Stanford, in a postdoc. What is that motivation factor that you can extend to our students, for this generation students who want to pursue the cardiovascular study, and how can they pick up the motivation from you?
See, as I said, it's the first thing in research. Research is really hard. Okay, that's the first thing that I would say. Research is like very. See, I'm still like studying basically, right? So it's a long journey. It takes like doctor, becoming a doctor and special, like achieving specialization is a long journey. So the first thing that people one has to understand is like, do they really want to do it, right? So research is like driven by passion, nothing else. The reward is very low, but it has to be driven through, only through passion. So if you really, really like research, and that's the first thing that they have to, ask themselves. The first question they have to ask themselves is like, do they really, really like research? The second thing is, especially for young people, maybe because it doesn't have to be like a cardiovascular research. So any kind of research. So it's a developing interest and then, working towards that. So as I said, I had planned for everything. Like, okay, I want to do a master's and then do a PhD and then do a postdoc and then become a PI. So it's like I had a plan that I set. So they need to have this kind of plan as well to if they want to, pursue research as a career. And other option, good thing is people who are in India. So I didn't make it to IITs, okay, IITs or the CSIR labs. People who wants to do research from India, IITs are really good places. If they make it to IIT and there are like labs, they do really, really good research. And when it comes to life sciences, CSIR labs, like, as I said, IASC, NCBS, JNCR, INSTEM. So there are like so many institutes. They are doing really, really good research in especially in like in biology, life sciences. So these are the institute that students should aim for getting in. I don't know I mean, it's so competitive in India. It's very hard to get into these. They have competitive exams that they have to clear, and then they will they have to choose these institutes. So a young person who wants to pursue really like good research, um, I realized this only after coming to the Canada, that they have something called as MD PhD. So you can do a doctor training at the same time you can do a PhD. That means you are getting trained to become a doctor to treat patients, and also you are getting trained to do research in the lab as well. So this is like a very good combination. I wish I had this opportunity when I was in like young, like I was in college. So IISc is starting this MD PhD program. I don't know how many students they will take. But these are like the opportunities that India has, and as far as I am aware of. So IISc is like starting this MD PhD, and CSIR labs is like they can aim after clearing GATE, NET, not NET, it's GATE, and there is one more exam which I don't remember. So these are the avenues or are the pathways that they can take to if they really, really want to excel in research. The other path that people take is like coming abroad. So coming abroad is a really, really good choice, I would say. I would still say that it's better choice than staying in India. I mean, people may not like it, but that's a reality. So the quality of research is really good, and also the research environment is really good. And when I say environment, it's the people, the mentor, the students, and in terms of the fellowships as well. I hear like many of my friends back in India saying that even though they have like all this NEET GATE fellowship, they don't give it on like every monthly basis. I don't know if it's still the case, but here it's, see, they have to understand, like students, we don't have like financial support, right? So here they give it to you like a salary. They don't delay it. So these are the things that I feel they are like very good here. And also the cutting edge research, especially in life sciences. Yeah, I don't think we can compare India to US research. India is like way far behind in terms of life science research. I can say that because it needs a lot of investment and also like huge infrastructure. So that India lacks that, and maybe like we are doing good. Hopefully, in another 10 years, we'll be like at a stage where we can do cutting edge research in terms of life sciences. I'm hoping for that. So yeah, that's the whole summary of my suggestion to the students who wants to pursue research as a career. So keep in mind it's a long journey, and if you do research only if you like, just because you want to have a PhD, call to be just to be called as doctor, a PhD is not a worth doing, and it takes like 5-6 years of time, and it's a prime time of your life, so it's worth spending on something that you're really passionate about. I hope I answered your question. It's like, I don't know who asked this question.
Thank you. This
Yeah.
is Ramakrishna. Thanks, Ravichandra. Thank you very much.
Yeah. Any other
Suresh?
question?
Suresh? Suresh? Suresh?
Sir, am
Are
I?
you there?
Yes, sir. ಕೇಳಿಸ್ತಿದ್ಯಾ?
Shall we conclude the meeting?
Yes, sir. I request Mahesh sir to give conclude remarks about the session.
Thank you, Suresh. Dr. Ravichandran, where is he? Not seeing.
Yeah.
Suresh?
Yes, sir.
Yeah. Now I can see. Yeah. Dr. Ravichandran,
It
you
sounds so
have
good.
shared your sweet memories at your school and college.
Yeah.
Then your passion for science is really great.
Yeah.
I myself feel very proud that I was your teacher.
Thank you, sir. Thank you so much.
You have shared all your experiences, which made you to come out of the box to do interesting things and further to research in electrophysiology.
Yeah.
You have described the functioning of the ion channels, PQR waves, and so on, and use of B blockers and drugs for cardiac problems. Stem cell technology for cardiac problems was very clearly explained.
Yeah.
You have briefed about your postdoctoral studies on the same.
Yeah.
Nice to hear about applying artificial intelligence for the drug design here also, in your field also.
Yeah.
So your talk was very informative, very inspirational and motivational. So thanks for motivating the young scientists towards research in general as such. And you have finally given the tips for their success, how to move to research and how to explore there. So on behalf of Prajna Foundation, myself, the chairman, and on behalf of my team, I heartfully thank you. Thank you very much for giving your time, sharing your experiences, and giving all that information to us and motivating us too. Thank you so much. Thank you.
Oh, thank you so much, sir. I mean, it's really an honor. Like you calling me Dr. Ravichandra. Really, I realize that I have a PhD now. Thank you so much. Like it really makes me feel proud that thanks for the opportunity. I really appreciate. Although I still remember those biochemistry lectures. So yeah, thank you so much, sir. Thank you so much.
Thanks a
And
lot. Thanks a lot. Thanks a lot.
yeah, and thanks, Suresh. Thanks for asking me to give this a talk. Sorry, I mean I just try to be explain everything in layman words so that people can understand.
It was
Yeah.
Yeah, really,
it was
that was
very
very much required.
useful session indeed. Okay.
Okay.
So I also thank Mahesh sir, giving me opportunity to find a person in this particular scenario. So I thought let me have one session from Dr. Ravichandra. So that's what I know. We have to thank Mahesh sir for the giving
No,
opportunity.
you belong to Prajna team.
Yes.
You have taken the responsibility of giving such a good talk. So really, I myself, on my own behalf, I thank you so much for organizing this session. Thank you, Suresh.
Thank you, sir. Definitely, I'll be happy be part of Prajna Foundation, and also thank you very much, Ravichandra. So we'll
Thank you, Suresh.
continue.
Yeah.
We'll give some more talks on this. Weekly talk definitely will be there from Prajna Foundation, and also
Yeah.
you can suggest any other personality who can give motivate our young minds, also young research fellows in India. So especially we are founded related to student empowerment only. So towards that we'll work together and give some empowerment to the students. Thank you.
Definitely, Suresh. Definitely.
you.
I would love to be a part of it.
Yes.
Yeah.
Thanks.
So
Thanks to
you-
all the participants who participated. So hope this session was fruitful. Thank you very much. Thanks again, once again.